Welcome to the deep dive. Today, we're going into an absolute anatomical masterpiece, the human eye.
It really is.
And our only source is the incredibly detailed Gray's Anatomy. So our mission is pretty clear. We're going to take this dense technical blueprint and, well, turn it into something you can actually visualize.
Yeah, the goal is for you to be able to just close your eyes and mentally walk through the whole structure, the fluids, the neurology, everything.
Exactly, and the clinical side of it too. Okay, let's just jump right in. Let's start with the gross anatomy, the outer envelope.
Right.
If you try to picture the eyeball, it's not just one perfect sphere. It's actually made of two different spherical segments sort of stuck together.
That's a great way to put it. You have the big posterior part, the white of the eye, which is the sclera. That's what, over 90% of the surface?
It's a protective shell.
It's the protective shell, exactly. And then fused onto the front is this smaller, much more curved and totally transparent dome, the cornea.
And that distinction is everything, isn't it? The big sphere is for structure, for the muscles to attach to.
Mm-hmm. And the tiny front sphere, the cornea, that's your window. It's the main focusing element, really. The whole point of the eye is to form a perfect, sharp image on the retina, and that starts at the cornea.
Okay, so to hold that shape, the eye is filled with fluid, but it's not all one big chamber.
No, it's segmented. The huge space behind the lens is the vitreous chamber. It takes up about 2/3 of the eye's volume, and it's full of this thick, gel-like vitreous humor.
And then in front of the lens.
That's where it gets a bit more complex. The iris, the colored part, divides that little space into two smaller chambers. The posterior chamber, which is right behind the iris, and the anterior chamber in front of it.
And both of those are filled with a much more watery fluid.
Yep, the aqueous humor. It's constantly circulating, bringing nutrients to the front of the eye.
Let's go back to that outer coat for a second, specifically that transparent window, the cornea. Because light hits it first, it has to be perfectly clear. No blood vessels.
Avascular.
Yeah. But it's also responsible for about 2/3 of the eye's total light-bending power. I mean, how does it manage that? Being so strong, yet so delicate.
Through incredibly intricate layering. The cornea isn't one sheet. It's 5 distinct layers, each with a job.
Okay, walk us through them.
So on the very surface, you have the epithelium. That's your mechanical barrier, your defense. Then the bulk of it, the thickest part, is the substantia propria, or the stroma.
And that's where the transparency comes from.
That's where the magic happens, yes. Its collagen fibrils are incredibly small and spaced with such perfect regularity that light just passes straight through without scattering. It's an amazing bit of biological engineering.
So it's all about microscopic organization.
Yeah.
What about the deepest layer? I know the endothelium has a really critical job.
Absolutely. The endothelium is maybe the most important layer for keeping the cornea clear. It's a single layer of cells with active pumps that are constantly pulling water out of the stroma.
So it keeps it from getting waterlogged.
Exactly. If those pumps fail from disease or damage, the stroma swells up, that perfect collagen spacing is ruined, and the cornea it goes cloudy. You lose your vision.
And one last thing from Gray's on the cornea. It's packed with nerve endings.
Incredibly rich sensory innervation, which is why even the tiniest scratch can cause such intense, almost disproportionate pain.
Okay, this brings us to the plumbing system, the fluid dynamics. We mentioned that aqueous humor that fills the front chambers. Where does it actually come from?
It's actively secreted by the ciliary body, specifically the epithelium of the ciliary body, and it's produced into that little pocket of space we called the posterior chamber.
Right, the space behind the iris. So from there, it has to get out somehow. For you to visualize this, the aqueous flows from that posterior chamber, squeezes right through the pupil.
The hole in the middle of the iris.
And then it floods into the larger anterior chamber. But the critical part is how it drains out. And that happens at the junction where the cornea meets the iris, the iridocorneal filtration angle.
That angle is everything. And right in that angle is the filter, which is called the trabecular meshwork.
What does that look like?
Think of it like a tiny, intricate sponge or a sieve. The aqueous has to percolate through this meshwork. And then it drains into a collection channel that runs all the way around the eye called the canal of Schlemm. From there, it gets back into the venous blood system.
So what happens if that filter, the trabecular meshwork, gets clogged? Or if the angle itself just gets too narrow?
Well, now you're talking about rising intraocular pressure, or IOP, and that is the direct cause of glaucoma.
Because the fluid can't get out as fast as it's being made.
Precisely. And that sustained high pressure looks for the weakest point in the eye's structure. That weak point is at the very back of the eye, a perforated plate in the sclera where all the nerve axons exit. It's called the lamina cribrosa sclerae.
So the pressure pushes on that plate.
It forces it to bow outwards. And in doing so, it physically shears and crushes those delicate retinal ganglion cell axons. That's what causes the irreversible blindness in glaucoma. It's a mechanical failure.
A heavy reminder of how important that pressure balance is. Okay, let's peel back that sclera now and look at the layer underneath, the middle vascular layer, the uveal tract.
Or just the uvea. It's one continuous layer, but we describe it in 3 parts. The choroid at the back, then the ciliary body, and the iris at the front.
Let's start with the choroid.
The choroid is basically a thin sheet packed with blood vessels and pigment sitting right inside the sclera. Its only job, really, is to be the nutritional supply for the outer layers of the retina, which don't have their own blood supply.
So it's a dedicated life support system.
It is. Graves describes its layers with larger vessels on the outside Haller's layer feeding into smaller vessels inside Sadler's layer. And all of that ends in the choriocapillaris, a super dense, super permeable capillary bed right up against the photoreceptors. It allows for incredibly fast delivery of oxygen and removal of waste.
Okay, moving forward from the choroid, we hit the ciliary body. We already know it makes the aqueous humor, but it has a mechanical job that's just as important. It's how we focus. Accommodation.
So to visualize this, the lens is suspended by these tiny tension wires, the zonular fibres. They stretch from the lens to the ciliary body. How does the muscle there actually change our focus?
It's a bit counterintuitive. The ciliary body contains a smooth muscle ring. Now, when you want to look at something up close, that muscle contracts.
Okay.
But instead of pulling things tighter, the contraction actually moves the entire ciliary body forward and inward toward the lens. This movement relaxes the tension on those zonular fibers.
And that lets the lens change shape.
Exactly. The lens is naturally elastic, so with the tension gone, it springs into a rounder, more convex shape. That increased curvature gives you the extra focusing power for near vision. That whole process is accommodation.
Accommodation is always paired with the action of the iris, right? The
Yeah.
eye's adjustable aperture. The diaphragm of the camera, yes. It controls how much light gets to the retina, and it does that with 2 tiny muscle groups.
Tell us about those. How does the pupil constrict and dilate?
So the sphincter pupillae is a circular muscle, like a purse string, right around the pupil. It's controlled by the parasympathetic system. When it contracts, the pupil gets smaller. That's meiosis.
And the other one?
The opposing muscle is the dilator pupillae. It's a thin layer of fibers that radiate outwards, like spokes on a wheel. It's run by the sympathetic system, your fight or flight, and when it contracts, it pulls the pupil open. That's mydriasis.
And these muscles are part of some key reflexes.
Oh, absolutely. The pupillary light reflex is a huge one for neurologists. You shine a light in one eye, and the signal goes to the brainstem, which sends signals back to both eyes to constrict. That direct and consensual response tells you a lot about the brainstem's health.
Okay, let's talk about the lens itself. The centerpiece of that focusing system. A really striking detail from the anatomy is that the lens never stops growing.
That's the key right there. New cells are continuously laid down your entire life. So the lens gets thicker and bigger over the decades. And what's fascinating here is that this continuous growth is, well, it's pretty much the main cause of presbyopia.
The need for reading glasses as we get older.
Exactly. As the lens gets thicker and less flexible, the ciliary muscle just can't generate enough slack in those zonular fibers anymore. The lens can't get round enough to focus up close.
And that thickening has other consequences too, doesn't it? It's not just about focus.
No, it's a space issue. As the lens bulges forward, it pushes the iris with it. This physically makes that drainage angle, the iridocorneal angle we talked about with glaucoma, shallower. That's why the risk for angle closure glaucoma goes way up into older people.
Let's ground this in the idea of a perfect eye, or emmetropia. That's where a distant image focuses precisely on the retina without any effort. So if the eye's total power is about 60 diopters, mostly from the cornea and the lens, what happens when the geometry is off?
That's when you get refractive errors. If your eyeball is a little too long for its focusing power, the image lands in front of the retina. That's myopia, or short-sightedness.
You can see close, but not far.
Right. And if the eyeball is too short, the focal point is behind the retina. That's hyperopia, or long-sightedness. And then, of course, if your cornea is shaped more like a football than a basketball, the focus is uneven. That's astigmatism.
Okay, last thing inside the globe is the vitreous humor, that gel filling the big back chamber. What happens to it as we age?
It liquefies. It's inevitable. The gel structure starts to break down, it shrinks, and it pulls away from the retina. This is what causes floaters. You're literally seeing little bits of debris floating around in the now watery vitreous.
Usually harmless, but not always.
Right, usually it's fine. But if the gel remains partially stuck to the retina while it's pulling away, it can create traction. And that traction can be strong enough to tear the neural retina, which can lead to a full retinal detachment, a true emergency.
So now we've arrived at the sensory film itself, the retina. The neural tissue that actually detects the light. It's famously complex, described in 10 separate layers.
Yeah, but we can simplify. You've got two main types of photoreceptors. The rods, there are about 92 million of them, are for low-light black and white vision. They're most dense in a ring just outside the very center.
And then the cones?
Then you have the cones, far fewer, maybe 4.6 million, but they handle our high-detail color vision. And they are packed into the very center of the retina in an area called the macular, specifically in a tiny rod-free pit at its center called the foveola. That's where we get our sharpest vision.
And to get that sharpness, the retina has a special architectural trick right there at the foveola.
Yeah.
It actually pushes all the other inner layers aside.
It does. It creates a physical pit so that light has an unobstructed path to hit those cones directly. It minimizes any light scatter. You even have specialized glial cells, the Muller cells, that span the retina and act like tiny fiber optic cables, funneling light right to the photoreceptors.
And the final output from all this processing comes from the ganglion cells.
That's right. Their axons are like the final data cables. They travel across the inner surface of the retina, and they all converge at one single point to exit the eye, the optic disc.
And since that's just a collection of wiring,
there are no photoreceptors there at all. That spot is the anatomical basis for our blind spot.
now once those axons leave the eye, they form the optic nerve. But the real neurological complexity starts as that signal travels back to the brain.
The visual pathway. The anatomy here is what determines everything about what you see and what you lose when there's damage. The most critical event happens at the optic chiasma.
The big crossing.
The big crossing. The axons that come from the nasal half of each retina, the half closer to your nose, have to cross over to the opposite side of the brain. But the axons from the temporal or outer halves of the retinas do not cross. They stay on the same side.
So what's the point of that arrangement?
It ensures that everything you see in your left visual field, everything to the left of your nose, gets processed by the right hemisphere of your brain and vice versa. After the chiasma, the signals synapse in the lateral geniculate nucleus, or LGN, and then project back to the primary visual cortex, V1, at the very back of the brain.
And that crossing is everything for diagnostics. You can map where a problem is just by looking at what part of the vision is lost. For example, what if you have a tumor, say from the pituitary gland, pushing up right on the middle of that chiasma?
You knock out just those crossing nasal fibers, and since those fibers carry the information from your temporal or peripheral visual fields,
you lose your peripheral vision.
In both eyes. It's called bitemporal hemianopia. It's the classic sign of a chiasmal lesion.
And if the lesion is after the chiasma, in the tract or the cortex,
then you get a homonymous contralateral hemianopia. You lose the same half of the visual field, say, the left half in both eyes, and sometimes, bizarrely, with a cortical stroke. The very central part of your vision might be spared, a phenomenon we call macular sparing.
So, to pull this all together, we've really covered three huge themes here.
I think so. We started with fluid dynamics, the balance of aqueous humour, and how a failure there leads to the mechanical damage of glaucoma.
Then we moved to the mechanics of the uveal tract and the lens, how they work together for accommodation, and how that system fails with age, leading to presbyopia.
And finally, we looked at the retina and the brain, the incredible specialization for high-resolution vision at the foveola, and then that critical rerouting of all that information at the optic chiasma.
It's an astounding piece of engineering. And it even corrects itself. We talked about how myopia is an eyeball that's too long, but here's a final thought for you to chew on. The eye isn't passive. During childhood, a process called emmetropization actively monitors the image clarity on the retina. It then signals the sclera to either speed up or slow down its growth to make sure the eyeball's length perfectly matches its focal power.
A biological self-correction mechanism.
Exactly. So the question is, how on earth does the body achieve that level of precise mechanical self-calibration? Just by interpreting how blurry an image is.
That's a deep one.
Thank you so much for joining us on this deep dive into the anatomical foundations of the eye. We hope you have a far clearer visualization of this complex organ now. Until next time, keep exploring.